So it's great that we know all of this about how dendritic cells take up antigen. But what's important to note is that the dendritic cells actually have different ways of taking up the antigen. This may occur via receptor-mediated phagocytosis, where the dendritic cell will recognize components of the bacteria or the pathogen and engulf it or swallow it up. This may also occur via macropinocytosis, which is a non-specific way for the dendritic cell to take up things in the extracellular environment. You can think of this as the dendritic cell taking a drink of its environment. Another way that this may occur is via viral infection. The virus will infect the dendritic cell directly, and now the pathogen and parts of the pathogen are right there for it to process. Alternatively, cross-presentation may occur, and this is a process by which a molecule or a pathogen is taken up via phagocytosis, but then the pathogen gets into the cytosol of the cell. And finally, it may occur via transfer from dendritic cells that came from the peripheral tissue onto the resident dendritic cells in the lymph node. So why should I care about this? Well, the way by which the dendritic cell takes up the antigen determines how the antigen is presented to T cells later on. You can see here that if a pathogen is taken up by receptor-mediated phagocytosis or macropinocytosis, parts of that pathogen, or the antigen, will be presented on MHC class II. In contrast, if the cell has been infected by a virus, is taken up by cross-presentation, or has received antigen transferred from dendritic cells in the tissue, these are all presented on MHC class I. So the MHC molecules, as I'm sure you remember, are responsible for presenting antigen to the TCR on the T cell. However, what's important to note about this interaction is that the TCR is not the only molecule that interacts with the MHC molecule. T cells will also express a co-receptor called CD4 or CD8, depending on the type of T cell. This co-receptor will also interact physically with the side of the MHC molecule, strengthening the interaction between the TCR and the peptide. This means that only T cells expressing the appropriate co-receptor will be able to recognize antigen on certain dendritic cells. So you can see here that because CD4 is only physically capable of recognizing MHC class II, only T cells expressing CD4 or CD4 positive T cells can be activated by a dendritic cell presenting antigen on MHC class II. Conversely, CD8 is only capable of physically interacting with MHC class I, not class II, and thus CD8 positive T cells are the only ones that will be able to be activated by dendritic cells presenting antigen on MHC class I. There's a pretty easy way to remember this, and I call it the rule of 8. You can see here that APCs expressing MHC class II will only bind and activate CD4 positive T cells. 2 times 4 is 8. APCs expressing MHC class I will only bind to and activate CD8 T cells. 1 times 8 equals 8. So if you ever forget or get confused about which MHC interacts with which CD molecule, remember the rule of 8. While this interaction is incredibly important for knowing whether the T cell is going to recognize the antigen, it's not enough to activate the T cell. This brings us to the 3 signals necessary for T cell activation. The first we've already talked about. T cells must see the antigen presented on the MHC peptide, and there must be binding between the co-receptor CD4 or CD8 to the MHC molecule. Even when this interaction occurs though, the T cell must receive the second signal. The second signal is delivered by a set of costimulatory molecules, collectively called B7, that will tell the T cell that it's okay to survive and keep going on with the activation process. The costimulatory molecules are actually B7-1 and B7-2. They signal through the receptor on T cells CD28. When APCs become activated, or these dendritic cells become activated, they will begin to upregulate B7-1 and B7-2. This ensures the immune system that there is in fact an infection and that an adaptive immune response needs to be mounted. When CD28 binds B7 and B7-1 and B7-2 on the dendritic cell, the T cell knows that it's able to survive and move forward. If the T cell only receives the first signal that we talked about, that interaction between the TCR and the MHC, but it doesn't receive the second signal, it will not become fully activated and instead will die. However, if the first two signals are successful, then the T cell will receive the third signal, which are cytokines produced by the dendritic cell that will tell the T cell what kind of subset it needs to become. This is something we're going to talk a lot more about in the next lecture, so I'm not going to address it today. This brings us finally to our last objective for today, which is to discuss different co-stimulatory molecules. So we've talked a lot about different receptors on the surface of T cell and the antigen-presenting cell that are important for activating the T cell. However, what I told you is not all that there is. There are actually even more co-receptors that are responsible for supporting the activation and the proliferation of the T cell. Some of these include CD40 and CD40 ligand. CD40 will help upregulate the expression of co-stimulatory molecules B7-1 and B7-2 on the surface of the dendritic cell. You also have CD2 and CD58. These are responsible for initiating that first interaction between the APC and the T cell. IL-2 and IL-2 receptor are another important pair. IL-2 is the main proliferative cytokine that T cells see after being activated. Without IL-2, T cells can't proliferate and mount an appropriate adaptive immune response. These other molecules down here fall into the family of co-receptor or co-stimulatory molecules. These, in addition to B7-1 and B7-2 and CD28, either downregulate the activation of T cells or drive the proliferation of T cells. We already talked about CD28 and B7-1 and B7-2 when we talked about the 3 signals necessary to activate a T cell. However, today we're going to focus a little bit more on CTLA-4. CTLA-4, in contrast to CD28, is a coinhibitory molecule. This means that when it signals through B7-1 and B7-2 on the surface of the T cell, that it downregulates its ability to activate and proliferate and instead causes it to stop doing its activity and its effector function. CTLA-4 is typically expressed on the surface of T cells once they have been activated for a very long time. You'll see in a second why this is important. So now the question becomes, well, what if both CD28 and CTLA-4 are expressed at the same time? One of them has to win in the interaction with B7-1 and B7-2 to deliver the appropriate signals to the T cell. It turns out that CTLA-4 binds B7-1 and B7-2 on the dendritic cell with a much higher avidity. Note that I didn't say affinity. Affinity is the strength by which 2 molecules interact at one contact point. Avidity is an assessment of how many molecules one receptor can bind at one time. You can see here that CD28 binds B7 and B7-1 only at one time. In contrast, CTLA-4 binds multiple B7 molecules at the same time. This stronger avidity of the interaction means that CTLA-4 will always outcompete CD28. So if a T cell is expressing both CD28 and CTLA-4, it will not become activated and instead will be inhibited from becoming activated. So what are some important functions of CTLA-4? Well, CTLA-4 is expressed on T cells after TCR signaling and it plays an important role in regulating the immune response. Once you activate an adaptive response, you can't have it go on forever, so CTLA-4 is helpful in downregulating that response and putting you back at your normal homeostatic state. CTLA-4 also defends against autoimmunity by constraining inappropriate proliferation and activation of T cells. Sometimes our selection of T cells that recognize antigens that are only foreign doesn't work so well. This means that occasionally a T cell will become activated in response to a part of our own body. We can't have that happen. That would be really dangerous for us. So instead of becoming fully activated, CTLA4 makes sure that the response of these T cells is dampened and that we don't start attacking our own body. A third important role for CTLA4 is that it's expressed highly on regulatory T cells. I won't say too much about this today because it will be addressed in later lectures, but it's also important for dampening the immune system after it has been activated. And finally, it serves a detrimental but significant part in controlling tumor growth. This is something you'll hear a lot about later on when you learn about tumor biology and cancer. This brings us to the end of our lecture. However, before I finish, let's go through and revisit some of those objectives we talked about in the beginning. Our first objective was to introduce secondary lymphoid organs and sites of lymphocyte activation. This includes the spleen, which is located very centrally and is important for identifying blood-borne pathogens. The lymph nodes, which are located all over the body and are important for identifying pathogens that are infecting peripheral tissues, as well as the Peyer's patches. Peyer's patches are located in the gut and are important for recognizing antigens that come in through the intestine, but that's something we didn't touch on too much today. We also learned that secondary lymphoid organs are highly structured and are separated into B and T cell zones, where the lymphocytes can interact with specialized dendritic cells and decide whether they are going to become activated or not in response to a specific antigen. Our second objective was to learn about lymphocyte homing and migration to the secondary lymphoid organs and the chemokine molecules, as well as the surface molecules that are important in that process. We learned that L-selectins expressed on T cells interact with molecules like CD34 and glycam-1 on the surface of the endothelial layer to begin a rolling process that stops it from flowing freely through the bloodstream. We also learned that this rolling triggers the expression of integrins like LFA1 on the surface of the T cell. These integrins then interact with molecules like ICAM1 on the surface of the endothelial layer that will cause it to adhere to the endothelial layer and stop its rolling action. We know that once the rolling action has stopped, the T cell is able to migrate through the endothelium in a process called diapedesis, and then the T cell follows an increasing concentration of CCL21 and CXCL12 towards the lymph node. In our third objective, we learned about the 3 necessary signals to activate a T cell. We know that after entering the lymph node, T cells will interact with dendritic cells to survey potential pathogens. We also know that MHC molecules present antigen to CD4 positive T cells, and MHC class I molecules present antigen to CD8 positive T cells. And this is determined by how the dendritic cell picks up the antigen. We also learned that in addition to the TCR, CD4 or 8, and MHC interaction, the T cell must receive a secondary signal, a pro-survival signal delivered by the molecules B7-1 and B7-2 on the surface of the dendritic cell that signal through their receptor CD28 on the surface of the T cell. Finally, we learned that the third signal is cytokines that are secreted by the dendritic cell and tell the T cell what kind of cell it needs to become to address the specific infection at hand. Finally, we discussed different co-stimulatory molecules. We learned mainly today about CTLA4, which is a co-inhibitory receptor expressed on the surface of T cells. It, like CD28, binds to B7-1 and B7-2. However, it binds to B7-1 and B7-2 with a greater avidity than CD28, meaning that when the two of them are together on a cell, CTLA4 will win and inhibit the activation of T cells. For our next lecture, we'll talk about what happens after the T cells have become activated. We'll address the different T cell subtypes as well as their different effector function. Answering the question, what can an activated T cell do?